All right, let's get right into it. Today, we're going to be dissecting Pseudomonas putida, a bacterium that's, well, it's making a serious name for itself in our hospitals. Now, all the information we're covering is sourced from a Gemini deep search, and if you want to dig into the primary literature yourself, you can find all the references in the linked notebook.
Wow. Okay, this quote really just hits the nail on the head, doesn't it? It perfectly sets up the entire story we're going to unpack today. You've got this organism known for cleaning up environmental messes, and now the landscape has completely shifted. It's become a major opportunistic pathogen. That is a huge change.
So let's start right there. How did this happen? We're going to begin by digging into how this bacterium, once hailed as an environmental workhorse, has become such a big concern inside our hospitals.
And, a huge part of that story is its identity crisis. So we've got to talk about taxonomy. Because with the Pseudomonas putida group, getting the name right isn't just academic. It is a massive challenge. And getting it wrong, well, that can have some really serious consequences for patients.
Just think about how our identification methods have evolved over the years. We started with the basics, right? Looking at morphology, running a bunch of biochemical tests. Then we got more sophisticated with 16S rRNA sequencing. But here's the catch. For the Pseudomonas putida group, that 16S gene is it's too conserved. The species are so closely related that it can't reliably tell them apart. That's why today, if you want the real definitive answer, the gold standard is whole genome sequencing.
So this brings up a really important question for anyone working in a clinical lab. Is your ID really telling you the whole story? I mean, think about it. You get that MALDI-TOF MS report back that says Pseudomonas putida. You report it out, but what if that's not the full picture? Because MALDI-TOF is looking at protein profiles, the differences between species like, say, Pseudomonas alloputida and a true Pseudomonas putida can be incredibly subtle. Standard databases can easily miss them, and if that's happening, we could be completely masking the true epidemiology of an outbreak. And that really brings us to a critical point. When the stakes are high, we're talking severe infections, suspected hospital clusters, or a really nasty resistant isolate from this group, you absolutely need to go further. Whole genome sequencing becomes more than a research tool, it's an essential public health instrument for getting the speciation right and mounting an effective response.
Okay, so enough about genomics for a minute. Let's get practical. What does this bug actually look like when you've got it on the bench in the microlab? All right, so on the bench, it's your classic non-fermenter. You're looking at a gram-negative rod. It's motile, thanks to its polar flagella, and it's an obligate aerobe. It absolutely needs oxygen. It's happiest growing around 30 to 35 degrees Celsius, and yeah, it produces that characteristic fluorescent pigment, pyoverde, which gives it that yellow-green glow. But, and this is a key point, it does not produce the blue-green pyocyanin that just screams Pseudomonas aeruginosa. And that brings us to the money slide for any bench microbiologist, this right here. This simple panel of tests is your bread and butter for telling Pseudomonas putida apart from Pseudomonas aeruginosa. The big one, growth at 41 degrees. Pseudomonas aeruginosa can take the heat, Pseudomonas putida can't. That, plus the lack of pyocyanin, no gelatin hydrolysis, and no denitrification, it's a pretty reliable way to make the call.
Okay, so we know how to identify it, but what does that actually mean for the patient? Let's make that jump from the bench to the bedside and talk about the kind of diseases this thing actually causes. Pseudomonas putida is, well, it's the ultimate opportunist. It is not going around infecting healthy people on the street. It's what we call a pathogen of device and debility. And that phrase says it all. It needs two things to really get a foothold. First, a failure in the host defenses, almost always an indwelling medical device. And second, a host who is debilitated, someone whose immune system is already compromised. It preys on the vulnerable. So when you look at the infections it causes, it's basically a greatest hits list of nosocomial problems. We're talking catheter-related bacteremia and sepsis, hospital-acquired and ventilator-associated pneumonia, catheter-associated UTIs. And in the most fragile patients, you see devastating neonatal sepsis. This is a story that is completely intertwined with modern invasive healthcare. And this is why we have to take it so seriously. 39%. That's the 30-day mortality rate reported in one study of hospital-acquired Pseudomonas putida bacteremia. Let that sink in. That's not just a statistic. That's almost 4 out of every 10 patients. And it gets even worse when you're dealing with carbapenem-resistant strains. So no, this is absolutely not some harmless bug from the environment. So that's the problem. How do we fight back? With resistance on the rise and our patients being so vulnerable, how in the world do we treat these infections effectively? Here's the thing. There are no specific guidelines just for Pseudomonas putida. So treatment really comes down to two foundational principles. Pillar number one, you must be guided by in vitro susceptibility testing. Trust your lab's results. Pillar number two, source control. This is absolutely non-negotiable. If there's an infected line, a catheter, any kind of device, it has to come out, period.
So what does that look like in practice? Well, if we look at the Dutch SWAB guidelines, they provide a pretty good framework. Since they don't have specific data for Pseudomonas putida, they basically extrapolate from what they know about Pseudomonas aeruginosa. So, for suspected sepsis, they might recommend a combo like ceftazine plus tobramycin while they're waiting on those susceptibility results. Now, for kids, the principles are the same, but it's a whole different ballgame. Dosing is all weight-based, and you have to be so careful about antibiotic choices and toxicities. That's definitely a situation for a pediatric infectious disease specialist.
Okay, so for susceptibility testing, labs are using the EU CAST breakpoints for Pseudomonas species. But here's where it gets really interesting and where smart clinical microbiology comes into play. It's all about that I category, susceptible increased exposure. For so many of these tough-to-treat infections, the answer isn't always to switch antibiotics. The answer is to optimize the one you have. That means pushing the dose higher or using extended infusions for beta-lactams to keep that drug concentration above the MIC for as long as possible. That is a game-changing strategy for these bugs.
So why is this organism so hard to kill? Well, it comes preloaded with some serious defenses. Intrinsically, its outer membrane is like a fortress, and it's armed with these incredibly robust efflux pumps, things like TGBC, that literally just grab antibiotics and spit them right back out. But that's just the beginning. It can also acquire new weapons through horizontal gene transfer. And we're talking about the big guns here, carbapinamases like VIM and NDM, that can make our most powerful last-line antibiotics totally useless.
All right, so that brings us to our final section, infection prevention and control. And honestly, this might be the most important part of the whole discussion, because the best way to treat these infections is to stop them from ever happening in the first place. And this is where the strategy for Pseudomonas putida is fundamentally different from something like MRSA. With MRSA, we're focused on the patient, but for Pseudomonas putida, the fight isn't with the patient, it's with the hospital environment itself. Its primary reservoir, its home, is the hospital's water system. We're talking taps, drains, showers, that's where it lives. So an effective IPC strategy has to be multi-layered. Of course, you have your basics. Strict hand hygiene, standard and contact precautions, those are always essential. But for this bug, the real focus has to shift to the environment. That means rigorous decontamination and, most importantly, a really comprehensive water management plan. In those high-risk areas, your ICUs, your neonatal units, this can mean going as far as putting point-of-use filters on taps and showers, literally stopping the organism right at the source. And that leaves us with one last kind of provocative thought to chew on. We know Pseudomonas putida is perfectly happy living in the environment where it's exposed to all sorts of resistance genes. And we know it's incredibly good at surviving in the hospital. So what if it's not just another pathogen? What if it's acting as a key shuttle, a vehicle that's picking up resistance from the environment and ferrying it directly into our hospitals, delivering it to our most vulnerable patients? It's a pretty sobering idea. So what about you? Have you come across Pseudomonas putida in your lab? What's your experience been? We would absolutely love to hear about it, so please share your thoughts in the comments below. And of course, if you found this helpful, don't forget to subscribe for more explainers just like this one. Thanks for tuning in.